It's a delight for me to introduce our speaker for our keynote, Diego Restreppo, and I'm happy and excited to be actually doing this and introducing him because he's going to be talking about cerebellar circuits. And the cerebellum for so many years has been such a Cinderella of the central nervous system. There are more neurons there than in the cerebral cortex. And yet, all of us, me included, have focused on the cerebral cortex. So it's going to be really interesting to hear about these cerebellar circuits. The cerebellum has very, very interesting and idiosyncratic circuits. And Diego is going to be telling us about decision-making and activity in cerebellar circuits. So without further ado, Diego, it's all yours.
Thank you. And good morning from Denver. And I'd like to thank you. And it's wonderful to be in Neuromatch. And so let me start by sharing the screen. And I'm going to be talking about decision making. So hopefully you see the screen. And what's important here is, so this, by the way, started for a sabbatical. I was doing a sabbatical with a little microscope that you see here that Emily Gibson and I generated. And I went to Isabel Llano's lab. And I usually do work on olfaction in the front of the brain. And I became extremely, well, I was always extremely interested in the cerebellum because of what Cajal had done and a lot of people have done. And we started doing experiments and they became really, really interesting. So let me talk about this. And it has to do with go-no-go split decision making. And I'll explain what that is in a moment. And the work, of course, the important thing is the people that did it. And it was done by Mig Ma, who is a neuroscientist in the lab, Greg Futia, an engineer, and Beris Osbey, who helped us with the microscope as well, another engineer, and Fabio Simões de Souza from Brazil, who helped with modeling the cerebellum. So, and it's funded by the National Science Foundation and the NIH. So let me explain to you what go-no-go is. And I'm going to show you Roberto Clemente, who was playing for the Pittsburgh Pirates in 1971. And I'm going to show you a very quick movie, 3 seconds, on purpose. And the important part is part of the 3 seconds. So let me just start the movie. And I'm going to turn off the volume. And it's only 3 seconds that I want to show you. So I'm going to start here at 1 second and take a look at him hitting a home run. He hit 3 home runs in that game. And so he's looking at the pitcher and then hits the home run. Okay, that took place in a fraction of a second. And it's a very difficult decision, not just because you have to process the visual input, but because you have to process the history of what the pitcher was doing, what you're thinking, how you're feeling. And so it's a very difficult decision. And the cerebellum is involved in it. I'd like to call your attention to work with athletes, elite athletes, that was done by Owens et al. So I'm going to go to the next slide. And this is work where they worked with elite athletes, making split decisions, split second decisions. And in cerebellar cortex, they found that in the medial and lateral cerebellar networks, there's prominent activity during the fast feedback stage of the task and before there's any movement. And so that calls our attention. That's a go-no-go decision. Is the ball in the strike zone or not? So that kind of decision, of course, is done in the cerebellum. This is from Ramon y Cajal. This is the output cells, the Purkinje cells. And then we have the input, the sensory motor input, all the way through the mossy fibers, through the granule cells. And we're going to be focusing on these molecular layer interneurons. So we're going to look at those during the go-no-go task. Now, in our case, the go-no-go task is done with a mouse. And so I'm going to show you the task. So this is a mouse that is thirsty and starts roaming around the cage. And pretty soon they find that if they lick here, they get water. And then we start giving them 2 odors, 1 of 2 odors. And they have to lick for the rewarded odor. So that's the go-no-go in this case. That's hitting the home run. So you'll see the mouse coming in, mouse licks, the odor comes in, that is the right odor, and they have to stay there for 2 seconds and lick, and then they get the reward. then the mouse is going to come out and we'll have the unrewarded odor. And in that case, they do start leaking, but then they stop leaking very, very quickly. So what we wanted to do is, initially it was just because I wanted to record from the cerebellum since I was going to Isabel's lab, but very soon it became very interesting. What we did is we took PV-CRE mice and we injected a virus expressing GCAM6 or GCAM7, and we recorded from the lobule 6. And that was because Giovannucci and Wagner had talked about neurons in this area, in that case, granule cells, responding to reward. So basically responding in a cognitive task. And also Rowan et al. Had suggested that these neurons gate for learning with valence. They actually respond to valence. That was a suggestion. So again, this is the input, sensory motor input, goes to the granule cells, and I'm going to be talking about these molecular layer interneurons that then inhibit the Purkinje cells, the output cells. So let's look at what happens. Now, this was now done in a head-fixed animal. So on the left side, you see the head-fixed animal, same task, the Gonogo task. And now we have odors A and B. The A is the rewarded odor. If they lick, and that's a dry lick, by the way, they lick for these two-second segments, then it's a hit and they get water. If they do not lick, it's a miss. And then for odor B, that's not rewarded. If they lick, it's a false alarm and they do not get water. They actually get a delay in the next trial. And if they do not lick, it's a correct rejection. Now, what happened with the molecular layer interneurons? So you see now the field of view with a 2-photon microscope. And by the way, there's two types of cells, the stellates and the baskets. We went more superficial, so these are stellate cells. And what you see here is the response of those during the trial. So you actually see a large change in calcium, in GCAM6 fluorescence. And when we analyze this, we use actually something that's called Kaiman from Giovannucci et al. It's a constrained non-negative matrix factorization. And what that does is it actually pulls out the components that are independently firing, independently changing calcium in that field of view. When we do that, then we find the following. This is now a time course, and I'm only showing a subset of the regions of interest of the components. At the bottom, I'm showing the leaking. So whenever there's a little spike there, there's a leak. And these red vertical bars are when the rewarded odor was delivered. The blue ones are when the unrewarded odor was delivered. This was a mouse that was already proficient. So they had learned to respond to the rewarded odor. And what you see is really interesting. Basically, when you look at the rewarded odor, the red ones, the animal licks, of course, and there's an increase in calcium. So, and that happens in all of them. And when the animal receives the unrewarded odor, except for a small number of times, like in this time they actually responded, most of the cells do not respond at all. So we became very interested in this. This was initially just an experiment to go to Isabel's lab, but then we became interested in this. And we started asking, is this changing as the animal learns? So this is now trial number on the x-axis, and the y-axis is percent correct, and it's computed in a 20 trial window that shifts. So at the beginning, the animal is naive. They do not know which odor is rewarded. Then as time goes by, they figure out which odor they're supposed to respond to. And at this point, they're proficient. They're 80% or better. So they actually have been able to respond to the odor. What happens with the calcium changes in the MLIs? Now I'm going to show for each trial. So I'm going to show an average, and I'm going to show it for each trial. On the right side is an average. This is when the animal is naive. At the top, we have delta F over F. So that's actually the change in fluorescence in the GCAM. So calcium goes up, the fluorescence goes up. For S plus, which is the rewarded odor, or S minus, the unrewarded odor. And at the bottom is the leak frequency. So let's look first at leak frequency. This is the naive animal, and that is the proficient animal. So for the naive animal, the animal starts leaking. That's how they start the trial, by the way. They have to leak to start the trial. And then there's not much of a difference here. In fact, this is not statistically significant when the odor is given. This is the odor period. If you look over here to the proficient animal, the animal starts licking, the odor is given, and for the S plus, the rewarded odor, they keep licking. In fact, they increase the rate of lick, and then they get the water here. So, and they increase further. And if the odor is the unrewarded odor, they just stop licking. So very quickly, they make a decision there. Now, what happens to the calcium? What happens with calcium is that it's not significantly different between S plus and S minus when the odor is given and the animal is naive, but it becomes very different when the animal becomes proficient. So the calcium goes up when the rewarded odor is given, goes down when the unrewarded odor is given. And this is showing the same thing, but per trial, for each trial. And we did this, that was in one animal, and we did this in 5 different animals. And you see the results for the different animals. This is percent correct. So this is how well the animal is doing over there. And what you can see is that for the S plus, they increase, the calcium change increases, and it becomes significant. And for the S minus, it does not. So that raised the question whether this would encode for which odor we were giving the animal. Could we, from the activity of all of the neurons in the field of view, differentiate between the two stimuli? This is shown here, and it's shown at the top. So this is basically the coding accuracy. It's done with a linear deconvolution algorithm. So basically, we give the algorithm all of the inputs, all of the calcium at each time point, except for one. And then we ask, can we predict the one that's left out, placing a hyperplane in the n-dimensional space? So if this is the S plus and that's the S minus, we put a hyperplane in the middle. And what's shown here is this is the naive animal. This is, again, when the odor is given, that's when the reward is given. So the black one is odor. The red one is when the water is given. And when the naive animal comes in, the decoding accuracy is basically 50%. This area that's shaded, by the way, is just a shuffled area. So we cannot differentiate from shuffled. And then for the naive animal, when they do get the water, they, of course, make a decision. And from the calcium signal, we can actually differentiate between the two stimuli. This is very different when the animal actually learns. So this is the proficient animal. And you see that basically it's about 50% as it comes in. Then the odor comes in. And then very quickly, this rises above the shaded area, the shuffled. So there's a large difference we can actually decode. And this is shown on the right side, by the way. I'm going to move this out. Okay. On the right side, this is when the animal is learning, the naive animal. There's very difference. Well, there's actually no difference between odorant and shuffled. And when they are proficient, there's a big difference between odorant and shuffled. And of course, there's a difference at reward as well. So this was interesting. And it led us to look at at what was, there's a lot of different inputs to the cerebellum. There's sensory motor input. And one of the inputs that's been studied quite well is lick. And that animal is licking. So could this be just a motor reflecting the licking? And to look at this, we looked at 3 different phases. Pre-odor, before the odor. We looked at the phase where we have the odor in there. And then we looked at the reinforcement area. So remember, during the odor, there's dry licking. They have to lick, but there's no water there. In fact, it's water with sugar, by the way. And then during the reinforcement, there is wet licking where they do lick and they get the water and the reward. And this shows the delta F over F, and this shows the lick frequency. This is for one example. In this example, the lick frequency just increased slightly, but it didn't increase too much. And what we look at is there a correlation for the slope of lick frequency? So the slope of the lick frequency here, in this case, and the slope of delta F over F, so the slope of the calcium change. And what we found is that this is shown for one animal here. There's very little correlation during the odor and pre-odor, but there's a correlation for reinforcement. So the lick is actually quite correlated during reinforcement. And this is now the correlation coefficient, showing that for odor, it's basically around 0, and for reinforcement, it's pretty it's much higher. Then what we did is, of course, it's not just licks that are the input to the cerebellum. It's very a large number of motor and sensory inputs. And in this case, we looked at a subset of these inputs. So we did a generalized linear model fit to each of the calcium changes to try to see if we could predict. This is an example for a hit, that's an example for a miss, that's an example for a correct rejection. Could we predict the change in delta F over F from all of these inputs? And what were the inputs that we looked at? We looked at kinematics. We actually were doing a video of the mouse. We looked at the lick rate. We actually were detecting with a touch sensor the licks. We looked at velocity, acceleration, lick rate, derivative of lick rate. We looked at history also, the reinforcement history, the accuracy of the response, percent correct. And we also looked at the presence of the odor. And the black lines here show the fits for these examples. And so what we wanted to ask was, what percent of the variance can these different variables predict? So when we did this with the GLM, and we did it for all the animals, you see the results here. So it's basically the generalized linear model. And this is the kinematics. So this is, in fact, we put them all together. They each do about the same thing. They're not very different from pre-odor to outcome. They contribute for about 30% of the variance. So they do about the same thing. If you look at odors or licks, then it's totally different, and different in a really interesting way. So in terms of odors, they do not contribute at all before the odor. They do not contribute at all for outcomes, for the time when they get the reward. And they contribute about 80% when the odor is there. So it makes a large contribution to that change in calcium. In large contrast with the licks, it does contribute about 30% before the odor. When the odor comes in, it goes down, and then the licks actually contribute quite a bit during reward. So what this indicates, which is really interesting, is that these MLIs are actually responding to different things at different time points, which makes a lot of sense. Now, a question that came up is this something that is this a response to an odor, banana? Or is this a response to the stimulus that's rewarded? And what we did here is we reversed the reward. So we had one odor being rewarded. In that case, it was banana. It was isoamyl acetate, fake banana. And then mineral oil is the unrewarded odor. So that's basically no scent. There's a little bit of a scent in mineral oil. And what we do is, this is percent correct. We reverse that here. And of course, the animals make a mistake. They go backwards, and then they learn the game. And what we found was really interesting. Basically, what we found was, this is in 3 animals, is that the delta F over F, as I had shown before, is increased for the isoamyl acetate when it's a hit, when that's the rewarded odor. And it's a small change for the mineral oil. This is the change during odor. And it goes backwards. So this is a contextual, a response to the contextual odor. Is this odor rewarded or not? Now, a question that was important was to look at whether this really was licks or not. Even though we had done the GLM, we needed to be more careful about this. And this is work from Gaffield and Christie. And what they had done, this is done in Cruise 2, and Cruise 2 is well known to respond to licks. And what they had done is they had actually changed the reward. So they changed it from 8 microliters to 4 to 0. And what they then look at is the peak of the lick rate versus the peak of the change in fluorescence. And what they see is that there's a linear relationship between those two, because this is a response to the licks. That makes a lot of sense. So we did the same experiment, but in this experiment, on purpose, we did not reward one of the odors. We rewarded both odors 70% of the time. So this is not a go, no go task. This is a go, go task. So animal is at 50% if we just try to find out, are they responded to one versus the other one. And then we recorded the lick frequency. and we recorded the change in calcium with delta F over F. And the question was, is there a relationship between these two? And we changed the reward from 0 microliters to 32 microliters. And what you see is pretty interesting. So this is the dry lick when the odor is there. So they're just licking for what's going to happen in the future. And this is the wet lick when they get the water. And what we found is that for lick frequency, there is no relationship on reward volume during the dry licking, makes sense. But there's a very clear linear relationship when they are licking during the wet lick, when they're getting the reward. They find out, oh, I'm getting 32, I'm getting a lot. In contrast, for the delta F over F, there was no relationship between the two of those with reward volume. So either dry or wet change in delta F over F. That indicates again that this is not just a lick response. Now the question is, these MLIs are responding and we can actually predict which odor the mouse is being given, but what happens if we turn them off or we actually decrease their activity? And we did this with chemogenetics. So what we did here is we used CNO and we used DREADS, and we added HM4DI with an adenovirus. And then that HM4DI will inhibit the cells if you give them CNO. But of course, you have to do controls. And so for controls, we just injected a virus that did not have the HM4DI, and we expressed Emcee. And then for the experimental, we did HM4DI, and we did no CNO and CNO. And this is several sessions. So the animals naive first, and then they learn, and then after the next day, they learn again. So they're doing pretty well. Most of this is purple. This is Emcee with CNO, just to show that in this case, CNO is not changing the results. And the interesting thing, and this is HM4DI with no CNO, so they learn pretty well. But you see that there's a large change when we put CNO and HM4DI. So when we inhibit those MLIs, they have a hard time becoming proficient. And you can see that as an average here. Now, the cool thing, though, is that when we looked at the time course, it's shown here. So this is the frequency of licking. So this is the licking of the mouse. So they first lick at about 4 hertz, and then they come in. They have to lick a little bit to start the trial. And then if they get the rewarded odor, they lick a lot. And if they get unrewarded, they stop licking. And that decision is made fairly early in the trial. And this is shown here. And the interesting thing is that this is the control. So in the control, no matter whether we put CNO or not, there was a fast decision making. But with HM4DI, there was a much slower decision making. The dark blue is the one that's the one with the CNO and HM4DI. And in fact, there was a decision making that took several, it took about a little bit of like a second. Usually it's about 200 milliseconds. So inhibiting the MLIs, the molecular layer interneurons, led to a slowdown of that split-second response. So there was a differential response of the odorants by MLI interneurons that develops as the animal learns to differentiate the odors. The MLI interneuron responses reflect the contextual identity of the odorant. It's not just the odorant. The mice are slower in the decision if we inhibit the MLIs. And then we concluded that in the vermis MLI interneurons, they encode for the valence of the odorant. Thank you very much. If there's any questions, I'll be glad to answer them.
Hey, thank you, Diego. That was absolutely fascinating. And we do have a question from the audience. Actually, it's from Conrad Cording. And I was wondering, Yang, maybe if Conrad may want to come on stage and actually ask that question himself, because we do have the time today. We don't have a time crunch like in other sessions. So if Conrad is willing, he can, because he may have a follow-up question to his question.
Hi, Conrad. You're muted.
Yeah, he is muted. Oops.
Oh.
All right. Let me, while he's trying to get online, let me ask you what he was asking.
Sure.
He said, I am curious why the calcium signals look so massively correlated. When I look at spike trains, that usually looks very different.
I was really curious about that, and I do not understand it. In fact, we computed dimensionality, and the dimensionality was pretty low. And in fact, when we started doing that decoding with subsets of neurons, we could actually predict with a small number of neurons. Now, why is that the case? Is that because this is because of this task? So let me tell you. So the answer, of course, is I have no idea. Okay, but let me tell you what I think it is, and why we're doing something very different right now. That's a very simple task, the go no go task, where you're hitting that ball, and this is the one thing I'm doing right now. And so I think what happens is in that case, dimensionality goes way down because you're really interested in that. So what we're doing now, we're not doing that in the cerebellum right now. We're doing that in the olfactory bulb, but we're interested in doing it in the cerebellum. Is more complicated tasks. So the one that we want to do for the cerebellum, where I bet that dimensionality is going to go way up, and this is just me saying something, I have no idea if that's correct, is following an odor plume. Odor plume navigation is extremely different, very different, very difficult, because what happens is there's a turbulence, so the physics are different. And I bet in that case that we're going to have dimensionality that comes right up, and you still make decisions. But that's me just making up, to tell you the truth. I have no idea about that. So I bet
Yeah,
that
and
depends on the task.
yeah, and just picking up on your point about the picking up on the plumes, and there has to be a really good dynamic response in the system to be able to track that and to be able to
In history.
get
Yeah,
that. Hmm.
yeah, yeah.
So you're thinking that the circuit, so the information goes from the olfactory bulb straight to the cerebellar circuits,
So the
and
questions were yes. Yeah.
okay, and that's my question. And so then the question is, it kind of leads into the question I had. It's a follow-up question about them. Actually, let me check. There's another question coming in because
Uh-huh.
I may try and deviate you away from this discussion. But let me ask you. I'm going to come back to this. Let
Yeah.
me ask you a question from Furazan Farahani. Since we're having trouble getting people on stage, have you tried to do the same experiment with a higher rate of reward while blocking MIL? And in order to know if MIL is modulating the value of reward or it has a gating role somehow.
Yeah.
So
That
it's,
that we did not do, but that's an interesting question, and we have not done that, but that's important.
yeah, that's an important question. Thank you for asking that. So okay, and Conrad, okay, no, that's yes, we've answered those. Good. No, so okay. So let me get to the question I wanted to ask you about, and that is, and I started going there because we started talking about the circuit in the brain potentially for that, and so there are studies of obviously there are a lot of studies in prefrontal cortex with these kinds of tasks in many species, and so I'm going to put you on the spot.
Uh-huh.
And
Yeah.
I'm going to ask you, which is the cart and which is the horse?
Mhm.
So of those two important
Mhm.
axes, the cerebellar and the cortical axis, which one comes first, in your opinion?
Which one? That's a good question. And there I have to again think about what I'm thinking. I bet that it's different depending on the task, depending on how quick it is and what kind of decision you have to make. It might actually become cerebellar to supplementary motor, um, but very likely, prefrontal, of course, is involved. And it's so I think it depends on exactly, I come from physics, and when I came, I was totally naive about behavior. Goodness, the number of variables in behavior are amazing. And I think that what happens is that that's the case, that very likely it's different.
Yeah.
Where the response is coming from, likely from Pons, the auto response. Noam Sobel has a beautiful paper in humans where their cerebellar changes for odors. So clearly there's responses there. So
I just resonated with your comment because I studied biophysics before I went into neuroscience, so I understand exactly what you're talking about. But just I'm interested in because you made a comment about just in when you answered that question, which made me think of what you said earlier about it depends on the task, which makes a lot of sense. But I'm just wondering whether the type of task will also modulate the dimensionality of the system,
I
which is what
think
you
so.
were
So
referring to
in
earlier.
fact, that's a really good question. And it would be very interesting to make this task a faster task. So Dima Ringberg did that years ago with the olfactory bulb. Basically with this kind of a task, and also with alternative force choice, you can have it so that the animal can respond ASAP, uh, versus you have to, the animal has to lick for say 5 seconds, something like that. Things may be very different depending on what you're doing, and also changing the reward amount. So one and the other.
Yeah,
Um,
yeah, and the other thing, when you were talking about your experiment in the mouse, it made me think of two fascinating studies in macaque monkeys actually on metacognition in prefrontal cortex, and so that started me thinking along these lines. And one of those studies was by Yoshida et al., and the other one was by Middlebrooks and Summer et al. And they had two macaque monkeys, which they had trained for ages, and they had and both of these studies had pairs of macaque monkeys interacting with one another and observing. So it was like a task with a rule that there was. It was a go no go kind of situation where one person had to one animal had to do the respond while the other was watching. They both knew what the rules were, and not only did they examine the licking behavior for the reward in the monkey who was doing the task, but the monkeys took turns in doing it. The other animal had a response measured in what they were doing. So they were monitoring these errors, the partner's errors, if you will, and they were looking at activity in prefrontal cortex. It was absolutely fascinating. So my question to you now is, what would happen if you had two mice,
too much.
if it was possible to
Mhm.
even train and have a mouse who was watching
Mhm.
the conspecific doing the task if they had both learned it. What do you think would happen in an instance like that?
That's interesting. So one thing is that I don't know, and it would be interesting to see what happens. It would be something. It would be a little bit of a difficult experiment because they're really good at smell. So you would have to do it in two different places, which is fine.
Uh,
Um, I do not know. Did the monkeys get rewarded depending on what the other one would do?
well, they got the same. So if one got it wrong,
That's
the
really
other one
good.
didn't get the
Yeah,
reward
that's
either.
excellent. That sounds like a cool
Okay.
experiment with the mice. I do not know. I bet that it would be depend on which sensory inputs you give the mouse. Um, Even auditory, they can hear 20,000 hertz to 40,000 hertz. Could be really, really interesting. So actually, it's a really cool experiment.
Cool.
Mhm.
Well, I'll keep watching for your publications
Let's
for
do
when
it.
you actually do that experiment,
Mhm.
because I don't know. I'm a human experimenter. I don't understand much about the many differences between the brains of say monkeys versus mice, and so I don't even know if that would be an appropriate experiment to do because I don't know much. I know more about monkey prefrontal cortex, but I don't know if that would be
There's a,
those.
yeah, what you're saying makes a lot of sense. There's a big controversy about monkey versus mouse. Um, and there's some argument that initially that, oh, they're so just totally different. But then Schoenbaum came and showed many things that happen in prefrontal. And there's an argument that, oh, well, but they're different, but they are doing something alike. So I don't know. Um, Of course, we need to do experiments in different species.
Exactly. No, and that just underscores that we do that. Well, let me ask you then this, and this is again because it reflects my ignorance. Would that be an experiment that would be more likely to succeed in a rat?
So first of all, I'm more ignorant about. And my current thought is that the rat would be very different and interestingly different. But that's just the reason I started working with mice is because they can recognize each other by smell. They can recognize the MHC, two amino acids in the MHC, and the mouse, she will mate with the one that's different. Rats are predators of mice. They're very different animals. In fact, right now I'm interested in some ethologically relevant work. I don't know. Even if you try a wild mouse. So at some point we started some work with wild mice. They're totally different to a mouse. That for sure would be extremely different because these are congenic. We've been had them in the lab for 200 years or so. So that's not a mouse. It's a very different mouse.
So talk a little bit about what makes a wild mouse different. Is it their repertoire of odorants that they would be sensitive to? What would be the main differences that you would see between them?
So their environment is totally different, and they have experience with predators. So I think that it's a totally different animal. So I bet that the brain development is different. It's both brain and plasticity, development and plasticity. We didn't do a lot of work with the wild mice. We did some work looking at the anatomy. And the bulbs were different anatomically. So I would expect there's a difference in development.
So the bulbs were more, the bulbs were much more, I would imagine, differentiated in the wild type? Would that be the case?
It was just gross anatomy. So they were different in terms of gross anatomy. So I don't know much about exactly what the molecular differences were, but that would be really interesting. What's the receptors? What receptors are expressed, and what's the circuit like?
Right. Yeah. And I wonder to what extent this go-no-go cerebellar kind of mechanism is also influenced by one's ability to respond to threat in the environment, because obviously what you were talking about with wild type mice and their exposure to predators, I mean, there's going to be they're going to be the ones that are going to be freezing in the environment when there's a predator. And so I wonder to what extent there's an interaction between those threat circuits and the kind of go-no-go circuitry that you're talking about there. And I have no idea what I'm talking about. This is just something that I'm imagining.
That's a really interesting question. I would expect in the wild type mouse, we may see a very different behavior in the ones that survive. The ones that don't survive with the predator may have a freeze, and these other ones may escape, and that likely is some, I'm making it up, some plastic mechanism, some plastic circuit.
So where would you see that circuit would be? Would it be mainly?
So amygdala should be involved. I'm playing with amygdala lately, so BLA for sure, a prefrontal BLA. But that's again making it up.
No, and again, well, okay, that's okay because we're just speculating here. Does the BLA send connections to the cerebellum as well, and vice versa, that you know of?
Everything, I mean, most of it will come from pons, so it'll be not direct but indirect.
Sure.
And there is work on prefrontal, and I forget. I think Mimi and Luo was part of that, but I may be wrong. So yes, there is input to pons. So it would be input to pons. And I think that there's also amygdala to pons. I'm almost sure.
Oh, that's interesting. Yeah. That's interesting. Yeah, that's something that we haven't really explored even in the human brain. I mean, we're only starting to think about, cerebellar circuits and also amygdala circuits in the sense that people are starting to break down, particularly the amygdala, into its different nuclei, and which is, I think, really healthy, because I think that's keeping it as a unitary structure has been such a mistake for a long period of time. But I think that I find your work particularly exciting because it shows how the cerebellum can really influence highly sophisticated behavior. And that's something that people haven't appreciated. And, the sort of work that you described today, which was really, really elegant, makes you think of frontal cortex, the kinds of the data that you showed. If I was blinded to what you were showing and I didn't know what brain structure it was, I would have said you were in prefrontal cortex.
I agree. I was very surprised. When we started doing this, as I say, we were just going to Isabella's lab and we thought we should do some recording before we go there. And goodness, it was fascinating.
So do you anticipate then doing and I know this is difficult because this is a mouse, but is it possible to do recordings at the same time from, say, frontal cortex and from the cerebellum, do you think?
We've been doing some in hippocampus and frontal cortex. But yes, so in the future, I'm interested in that kind of a thing. How do these places talk to each other? And, which one is faster? And could it be that one is faster in one condition and slower in the other condition?
Yeah. And also as a function of task, because we were talking about that earlier, right? That would be really, really fascinating to see what kind of differences. What would you predict if you had a crystal ball with the kinds of tasks, and if you could record from the two structures, what kind of differences would you expect to see? Have you an idea?
It will not be, I bet so there's something else that makes it very difficult, which is if it were with a more maybe outbred population or something like that, you're going to see differences between individuals. And they might actually correlate with which area is responding faster, even in the simple task. But that's me making it up. Which goes back to your question of, if you're in the wild, what do you do when there's a predator? Do you freeze? And a subset, we're going to do that, and a subset of them are going to jump out.
Right. Well, it sounds to me like you have probably about 50 years work with this stuff. And you could do you're going to do some amazingly interesting studies and make us think about how important these cerebellar circuits are. And I would just like to thank you for such a stimulating talk and for putting up with my ignorant questions.
So thank you, Aina. And my questions are more ignorant, but that's what I tell my students. When they tell me something like that, that's the question I want. I had one student that did that, my first student, who solved something that was, it was beautiful, a beautiful PhD thesis from a quote unquote ignorant question.
Yeah, and that's what we've said here at Neuromatch. There is no such thing as a stupid question.
That's correct.
That's what we've said, and people have been very, very shy at this meeting about asking questions, which is unfortunate. But that is the case.
Yeah.
And Farazan Farahani says that there are similarities here, and visual-based decision-making are interesting. There are inhibitory inputs from basal ganglia that are really important to the superior colliculus. So there's another parallel there for you.
Yeah. The visual system decisions, I've always looked at those as well. Yeah, that's interesting.
Okay. Well, I think what we'll do is we'll wrap it up. And thank you for such an illuminating talk today.
Thanks for the questions.
Okay. And everybody, have a good rest of the day.
Bye-bye.
Bye-bye.